Okay, great, excellent. Okay, let's continue our class. So today we will talk about the new calculation and some talk about some new basic laws that are very fundamental but useful for us to understand the biology. Okay, to understand why the cell has this type of size, why it has this number of the cells in the body. So those are the purely based on the physical laws, but very useful. Okay, and then we will talk about the fundamental building blocks of the life following that. In terms of the announcement, I have one announcement here. So I uploaded the homework number 1 on the Canvas. All of you can start to work on that. It contains 3 questions. I do think the first 2 you can start to work on now. So they contain some very interesting calculation, and then you may find the conclusion or the results things counterintuitive. Okay, things that different from what you are thinking. So that will be useful. So the first 2 questions you can start to work on that. The third question in the homework number 1 should be based on the diffusion theory that we will talk about after we discuss about the building blocks. So if we have time, if I put the due date now extended to September 25th. If you still feel not comfortable to solve the problem 3 for the homework number 1 at this date, then you can just submit the solution for the first 2 questions. Then leave the number 3 for the next time. That's also fine. Okay. So after you learn more about diffusion, about the random walk, then maybe the problem 3 is a more appropriate for you to practice. Okay. Then at that time we will also give you homework number 2 that it contains the MATLAB coding and the more calculations there. Okay. So don't worry about it. Just go ahead to practice homework number 1. Okay. Okay. And at the end of this month, we will also have a midterm exam number 1 that will cover all the material we have covered so far. Okay. So use that as a practice opportunity to check whether you really understand what we discussed in the class or not. Okay. Don't worry about the score. This class is purely for the research, for you to we designed this class is for transformation of your brain, transformation of your thinking, so that it can hopefully it can help your future research. So don't worry about the score. Use all the homeworks and exams as the practice opportunities. Okay. Okay, that's all the new announcement I hope to share with you. So now let's start today's lecture. Okay, in last lecture, can everybody hear me? Yeah, excellent, good. So in last lecture, we discussed about the cytoskeleton, rest tension, and the membrane, and the force propagation velocity of the membrane due to a force, and they are the localized propagation due on the membrane due to the anchored transmembrane proteins, right? We also talk about that the multifunctionality of the membrane. It has the function in both in terms of the chemical, mechanical, and electrical manners, right? So membrane is very important. Now, from this lecture on, we will look into those different components of the cells in a more quantitative way. So firstly, we will do some calculation, useful calculations, and also learn some physical laws. So when we talk about the biophysics, actually there are the 2 really important axes we care about. One is the spatial axis, the spatial distribution, spatial dynamics of the entities. Second axis is the temporal. What's the temporal scale? What's the speed? What's the temporal dynamics of the molecule of the cells? So they together gives the spatial temporal dynamics. Okay. So in here, let's first talk about the spatial domain. So this picture shows the distribution from the smallest entity to the largest living system. Okay. So the smallest entity, such as the atom, of course, the electron and the proton are even smaller, but the atom is by itself is a functional small system by itself, right? So atom is about 1/10 nanometer, so it's a 1 angstrom atom, and then many atoms combined together, they can constitute molecules. There are small molecules such as the water molecule here. This is a water molecule. And so all the living system currently we discovered on this planet, they all of them need water molecule. Okay. And then there are also other molecule called a macro scale molecule. They contain thousands or millions of the atoms together, such as the membrane. So lipid. Okay. So this is a lipid. They contain this is actually one half of the lipid. So the circle means the hydrophobic end of the lipid. The tail here stands for the hydrophilic part of the lipid. So this is actually a lipid monolayer. For the cell membrane, actually it's the lipid bilayer. So two layers of this combined end to end combined together become a barrier of the cell. Okay. Also, there are many proteins, right? The big protein and small proteins, they all belong to macromolecule. The protein is a real functional machine in the cell. They do all the jobs in the cell, right? And then, but all four of them are not living. They are not living. They are actually combined together to become higher order system that are regarded as a living system. So cell is usually in the micrometer scale. So between the micrometer scale and nanometer scale, there is another entity called a virus. So we talked about this virus. They are not regarded as a living system, right? Even they have spatial temporal dynamics. And then for the cell, there are also different types of the cell. They have different size. Usually, cell is 1 micron to 10 micron in one dimension. Okay, and then for the other cells like the egg, fish's egg, or the other types of eggs, they are actually bigger. They are millimeter. They actually can be seen by the naked eye. So when people do the injection, do the transgenetics experiment, they inject the DNA into the egg, into a mouse egg or the fish egg. They actually do it by you can do it by naked eye. Okay. For the smaller entity like the atoms and the small molecules, if their size is less than 250 nanometer means the diffraction limit of the light, then light microscope cannot be used to see that. Then we have to resort to electron microscope. That also means we need to fix them. We need to fix all these items and then coat them with some metal coating surface. And then electron microscope can be used to see the structure of the atoms. Okay. So here, actually, one thing here for the virus, as I said, the virus can be seen by the electron microscope. Means virus is dead. If we want to see the spatial temporal dynamics of virus, in fact, now people can extend the light microscope. We can extend it to all the way up to 10 nanometer now. So at the 10 nanometer, all the material here, the protein, the virus, now we can see live things. We can see their dynamics in the solution. Okay. But for the even smaller one, we still cannot use light microscope. Okay. Now, if we go even larger to the area that can be seen by the naked eye, then we see the animal, small animal, big animal, or even huge animal. And the largest living system in one dimension, the largest one is the California redwood. This is the longest, tallest. Okay. others are not that tall. Okay. So among all from here, if you look at this wide distribution of the spatial scale, we may wonder why human being is at this size. Why when we consist of, because we know human being and other animals are all consist of many cells. Why the cells are small? They are all small in this micrometer scale. Why cells need to be in the micrometer scale? Okay, that's in one end, the smallest living system, and also in the largest system, in another end of the spectrum, for the largest animal. Why the animal cannot be even larger? So here is a 100 meter for the California redwood, but for the blue whale, the size is 10 meters to 100 meters in this range. Why they cannot be even larger? Can we add more cells into those animals to make it to be even larger? Is that possible? Okay, so those are the questions. When you look at the spatial scale, really naturally, you will start to think about it, right? So think beyond what is showing on the slides. Same question you can ask if when in the future we show the scale of the temporal scale, when the cell or the animal doing some functions, there is a faster process. Some process can be started and finished within 1 nanosecond. Some other process may take days, months, and years. Why the process is like that? Can it be even quicker or even slower? Okay. So those are the questions. There must be some law behind it to govern those processes. And the question is, what are those laws? How can we understand this process? Okay. So let's first explore those questions associated with the spatial scale of the life. Before that, one question I wanted to ask you first. That is, if we want to look at the scales, spatial scales of the cells and also of the human, how many cells actually each adult human contains in our body? How many cells do we have? Here I have a multi-choice question here, and I want you to tell me what answer you think would be. How many cells we have in our body? And then I will show you the calculation, one type of calculation. Okay, so will it be 1,000, 1 million, 1 billion, or 10 to 14th, or 10 to 16th? Which one will it be? How many cells in our body? Can you guys tell me?
I don't know exactly, but my best guess would probably be D, 10 to the 14th, because I believe we have over 1 billion cells in our body, but I feel like 10 to the 16th might be too many.
Okay, so you are guessing it. So you have some understanding there, and you guess it. Okay, that's
Yes.
very good. That's very good. Excellent. Any other guess? Any other guess?
I mean, it's just a guess, but I'm gonna go with 10 to the 16th because I know that I guess cells can fold as well. So first I was looking at 10 to the 9th, and if we look at the nanoscale, like a nanometer is 10 to the -9th. So 10 to the 9th cells, supposing each cell would be a nanometer in width, it would be like a meter of cells, and just knowing that they fold a lot and conform in complex ways, I think it could go really large. So E 10 to the 16th.
Okay, good. It's also good guess. But I think your calculation can be improved better. Okay. Good discussion. Very good discussion. Daniel, right?
Yeah.
Yeah. Excellent. Good discussion. Yeah. Any other guess? Good, good. So that's a very good discussion. I think most people will select these two. Okay, this is correct. A very good trend. So either D or E. Okay, because in our brain, so this is a billion, right? 10 to 9 is billion. You know how many? You know, for human, when all our thinking, all our emotion are coming from the brain. How many neurons in our brain do you know?
Oh, sorry. I know there may be 2 billion of cells in our brain.
2 billion cells in the brain.
Yeah, but for neurons, I think maybe less than 1 billion.
In fact, in the human, actually, we have a 10 billion neuron for human.
So for human or for human brain?
Human brain. Human brain.
Okay.
Yeah, 10 billion.
Thank you.
10
I see.
billion. Yeah. Tens billion. Okay, so the number can vary a little bit, but the tens billion. Okay, so the correct answer should be between these two. In fact, is D. It's mostly like D. Okay, so we can do a calculation to find out which one it is. So the way to calculate how many cells in our body, we can use the volume. We can use the volume to check. Okay, so for the adult person, if we calculate the volume of the person, so usually it's between 1 meter to 2 meters, right, in the one axis, and then other two axes are less than 1 meters, right? Then if you roughly estimate the volume of a human for the person who is 100 kilogram, around this range, okay, around this weight, the volume of the human is about 10 to minus 1 meter cube. It's about that, about this volume. For the cell in the body, there are big cell and small cell. Some cell like a neuron, they are little bit bigger, and also egg also a little bit bigger, right? But some other cells are smaller. So on average, if we average all the cell, every direction of the cell may be about 10 micrometer, or even a little smaller than 10 micrometer. Okay, so overall, if you calculate the volume, if we assume every direction is 10 micrometer, cell is in a 3D condition, okay, in 3D. So then it's about 1,000 micrometer square, 1,000, okay, about 1,000. Sometimes it can be bigger, some 1,000 to 10,000. So let's see 1,000 micrometer cube. So 1,000 micrometer cube, if we convert micrometer cube to meter cube, then it's 1,000 10 to 3 times 10 to minus 18, 10 to minus 18, right? Meter cube, okay? Then we can divide then this value divided by this value. So this together 10 to minus 15. So 10 to minus 1 divided by 10 to minus 15. So this will be around this value, 10 to 14, okay? Around this value. Of course, this value can vary a little bit, but eventually they will fall into the range 10 to 14, and the upper limit is about 10 to 15 is upper limit. 10 to 15 is the highest. Okay, so that's about 100 trillion cells in our body. 100 trillion. Okay, so Daniel, you are not too far away. You are in the correct trend. Just exceed a little bit. Okay, and call your estimation based on the size of other organs. I think that's good estimation. Okay, do everybody hear me?
Yes.
Yeah, excellent. This is a rough calculation. Okay, so we have about this number of the cells. Now the question is, my question is, we have so many cells in our body, each of us. Then why inside the body we have so many of those tiny cells? Why not every cell increase their volume so that this number can be smaller? So we have a cell bigger size, but this number can be smaller. Why not like that? Okay. So to understand that, actually, there's one law we need to understand. That law is called a scaling law. Scaling law. So scaling law, we can use scaling law to think deeper about why cells are always small. So let's look at the scaling law. So for the volume, for any volume, here I use a cube to represent a volume. Okay, cube. So we have 3 cubes. They have different edge lengths. 1 millimeter edge length is 1 millimeter cube, and then 2 millimeter cube and 4 millimeter cube. So we have 4 cubes. When you think about the cell or think about any living system, in fact, there is a very important metabolic activity. So all these entities, for example, cells, they need to live, they need to survive. So in the cell, when they perform metabolism, they actually have a balance. The food they eat should be equal or even larger than the energy they consume, right? So the energy from the food should be larger or equal to the energy they consume. Cannot be smaller, then they will shrink, then will die, right? So it should be like that. And then for the cell, the food they obtained actually is through their membrane, through their surface. So the energy they can obtain is proportional to the membrane. Larger membrane, more proteins on the surface can, like ion channels or pumps, can help to get the energy, get the ATP into the cell. So the energy they obtain is proportional to the surface area. And the energy they consume in the body, on the other hand, that is proportional to the cell's volume. So everywhere in the cell, in the body, in the volume, will consume the energy because they are performing the functions. Okay, so the energy consumed is proportional to the volume. So let's calculate what is the surface area and the volume for these 3 representative cubes. For the cube number 1 millimeter cube, the surface size is you have a 6 side, right? 6 side, 6 times 1 square, right? Each surface area is 1 square. So then we overall get a 6 millimeter square. For the volume is 1 cube, so 1 millimeter cube, right? So for this guy. For the second one, also 6 sides, but now surface area is the 2 cube, so become 4. 4 times 6, 24 millimeter square. For the volume is 2 cube, so then is 8, right? For the number 4, for the number 3, 4 millimeter cube, 6 side times 4 square. So 4 square is 16. 16 times 6 is 96 millimeter square. And then the volume 4 cube is 16 times 4, so it's 64 millimeter cube, right? Now, as we said, the ratio of the energy should be equal to surface area to the volume should be equal to 1 or equal to larger, so that energy can be insufficiently supplied to the cells. Now, if we calculate the surface area to the volume ratio for these 3, for number 1 is 6 to 1. Number 2 is actually reduced now, 3 to 1. As the size increase, it reduces. Number 3 is even reduced, 1.5 to 1, even reduced. What it tells us is when the single entity, when their volume increase, in fact, surface area correspondingly also increase, but increase at a slower proportion. And then surface area to the volume ratio actually overall is decreasing. Okay. If we represent any edge of this cube, the length as L, then for the surface area is 6 times L square, right? 6 sides, and then area L square. And then if we divide by the volume is L cube. So this together is 6 divided by L. So as we change the L to be larger because it's in the denominator, right? It's in the denominator. So if L increase, this whole thing will decrease. That's actually the why the cell size cannot be too big. If it is too big, each cell, the energy they obtained from the surface is not sufficient for them to maintain the function inside. So the big cell has to die. Okay. So as the size decreases, the surface area will increase. Okay. So that's the reason why we have so many cells in the body. Okay. Can everybody hear me?
Yes.
Yeah. Excellent. Excellent.
I have a question. Does
Please.
this law relate to almost like the natural lifespan of organisms? So does the size of an organism based on this law relate to its lifespan?
Good question. Very good question. Here, I'm not talking about the size of overall organism. I'm talking about the single cell. If the human grow up, they will add more cells to body. Each cell is by itself maintain an energy balance there. Okay. When you talk about the overall the body increase, that's another law has to be considered, which will be in the next slides. Yeah, but a very good question. This is excellent. Isabel, you have a question?
No questions.
No question. Okay, good.
Thank you.
Excellent. Okay.
I have a quick question.
Yeah, please.
Wouldn't increased surface area also potentially cause an issue if like the environment temperature or something like that is not favorable?
It's possible. Yeah. So then that has to that is involved into the other issues, right? Right now we are all talking about the constant temperature, right? If the temperature increase, of course it will the cell will be exposed to a higher chance to increase their temperature. But the membrane also has a mechanism to release the heat. There are other ways to release heat. Okay. So those mechanisms already been considered throughout the evolution process.
Okay, thank you.
But a good question. Yeah, I like you guys all ask questions and just shout out. You don't need to raise hand. Just shout out. Okay. Good. Excellent. Good. So and there's then let's talk about another one for the whole organism. Okay, whole living organism. Now, we talked about as we seen in that spatial scale, all the animal, the largest animal like that whale in the ocean, you see the size is 10 to 100 meters, 10 to 100 meters, right? But that whale is not living living on the land. They are living in the water. Okay, on the land there are no such bigger animals, right? After the era of the dinosaur, right? So why the animal on the land, the size do not exceed that 10 to 100 meters scale? Why not? People in the fiction, people designed a figure, right? The King Kong is very large, but why in reality? But the question is, in reality, can we expand? Can we magnify a regular Gloria, magnify it 10 times from 6 feet high to 60 feet high? Can we magnify the regular Gloria to the King Kong, and then it still survive there? Is that possible? Okay. So for those animals to be survived on the land, they need to walk, they need to run, they need to be dynamic. The weight of the animal is determined by its volume, But the capability of the animal to be able to run, to walk, actually is determined by the leg, The leg has to be strong enough to support the weight of the animal, and the leg, the pressure applied to the leg equals to force divided by the size of the leg, of the feet, right? Cross section of the feet. if given the same force, like the same weight applied to the feet, if the feet size is smaller, then the pressure is higher because area is smaller. Force intensity is larger, right? Force intensity is larger. If the feet is larger, cross-section area is larger, then the weight distributed there on the large area, makes the pressure also to be smaller. That enable the animal to be able to walk without damaging the feet, right? So if we magnify the gloria to be larger, then the volume will increase as the L cube, but the feet, the cross-section area will increase as the square, as the L square, length square, right? So mass will increase if we increase, if we magnify regular gloria 10 times, then the mass will increase 10 times, 1,000 times. But the cross-section area of the feet will increase proportionally to the cross-section area, right? Will proportional to 10 times 10, so 100 times only. Okay, so then the strength of the feet actually increase 10 times less than the increase of the mass. Okay, so then at some point, when the mass is too large, the strength is not sufficient to support this mass, not sufficient to support the weight, then this animal cannot walk, cannot run, cannot hunt for its food, then they cannot survive on the land, right? So this also means even before animal are not unable to find food, even we increase the size of some animal, in fact, the animal that is larger sized generally can also be weaker than the animal that is smaller. Okay, for the King Kong, proportionally, it is about 10 times weaker than the regular gloria. Okay, in the feet, actually there are also bones inside. So the bone is the major load carrier components in the feet. Okay, so the bone size determines how strong the animals are. Yeah, and also for the King Kong, if we magnify the regular gloria into a 10 glorious weight on her or him, then they actually couldn't walk that well. Okay? They couldn't go around that flexibly to hunt for the food. So that's also the reason that if we look at the gazelle and the bison, if you look at these two animals, the size of them is not 10 times different. Okay, their size is not that different. But if you look at the bone inside of them, gazelle's bone and the bison's bone, they actually are really different. They actually different. At the thinnest part, they actually have 10 times difference, thinnest part compared to the whole cross section area. Okay, so at the similar weight or just a few times different of weight, but compared to the 10 times different of the cross section area of the bone, if you combine that together, then strength of the bison is much higher than the strength of gazelle. Okay, so the bison is much stronger. Okay, now based on this, we can understand better why those whales, why those big whales prefer to live in the water. The whale, the size is up to 19 to 20 meters. Okay, human is only 1 to 2 meters. And then even for the animal, for the elephant, they are much smaller than the whale. Okay, now because whale is living in the water, water can help them to support the weight. The water can help the whale to float and then to flow, to swim around. Okay, that reduce the chance the weight of the whale crashes on bones. Okay, so this actually is the one major reason that the whale prefer to stay in the water and they can survive there. Okay, there's actually another mechanism. If you think deeper about this, if you use the scale law I talked about on the cell, you can, if you use that law into the whale, the metabolism, if you calculate the surface area energy, consumption, and compared to the volume, you will find also living in the water is preferred by the whale, because in the water is a much better heat conductor than the air. In the water, the temperature increase due to the metabolism. Actually the whale can increase a lot of temperature because it's larger body, okay, larger size, and then that bigger heat generated by its own weight, own mass, actually is not sufficient, cannot be sufficiently dissipated by its surface. But if the whale is living in the water, water actually helps them to remove the weight, remove the heat from the surface. Then the balance of the energy is maintained in the whale body, so that the whale will not die because the temperature increased too high. Okay, that's all. That's why water is preferred by the whale. It's just like in our computer. Okay, we can use the microfluidic water. You use the water in the microfluidic channel to remove the heat from the CPU, from the some kind of high power CPUs. Okay, so that's the same reason. Can everybody hear me?
Yep.
Yeah, excellent, good. Okay, now with those laws, when we think about when we look into the biophysical experiment, biophysical behaviors, we always need to have those laws bearing in the mind. Okay. Now let's look deeper into the cells, and we can start to talk about the building blocks of the life. So just like every building is made of the blocks. Every cell also makes of its own blocks. Okay, except the blocks for the cell are small molecule and the macromolecule. They have the size. They have the size from the submicrometer to the subnanometer. Okay. In the cell, if you group all the building blocks together, we will find there are essentially 4 types of building blocks for the cell. Is this things true on this planet? I'm not sure on the other planets whether this is true, that still holds true or not. But on the Earth, this is true. Okay? Those 4 essential macromolecule, we call it a macromolecule. They are DNA and RNA. Okay? DNA and RNA. One is DNA and RNA. Second one is the protein. Third one is the lipid. The first one is a sugar, or we call it a carbohydrate. For the DNA, this is the macromolecule. They are made. All these 4 macromolecules are made of small pieces, small molecules. Okay. For the DNA, they are made of nucleotide. So this one is DNA. A is DNA. This DNA. Okay? And then they form the double helix. Second one is protein. Protein is made of amino acid. So this one is a typical protein made of many amino acids. Okay, and then between DNA, RNA, and the protein, there are correspondence there. Right, we discussed that. Lipid. Number 3 is lipid. This is one lipid molecule. Okay, lipid is made of fatty acid. Okay, they generally are hydrophobic, but they also have a hydrophilic components there. Yeah, so made of fatty acid. For the sugar, that is a carbohydrate, they are made of a simpler sugar. So they are small version of the sugar, and then they polymerize together to form the bigger version. That is a carbohydrate. Okay. Now, in the cell, these four components, these four building blocks work together with the water to form the basic chemical composition of the cell. If we for the different entities, the water components almost all the same. If you see here, and when we compare the single bacteria to single mammalian cell, water components always about 70%. 70% are water molecules, and then the rest of them are made of those four basic building blocks. Their concentration, their arrangement can be vary a little bit. Okay, in the bacteria, they have DNA, they have less DNA compared to the RNA. For the mammalian, they have 1:4 ratio of DNA to RNA. Okay? And then rest is protein and the lipid. So here is the pie chart showing that the water components in any single cell, and then the rest of materials. Okay, for the molecules, they have these four components. And then these four components can actually be even further categorized. They can be separated into multiple types in each of them. Overall, each type is one building block. Okay, if we look at the nucleic acid, nucleic acid will constitute DNA and RNA. Nucleic acid, we actually have eight types, 4 for the DNA, 4 for the RNA. Okay? For the sugar, okay, carbohydrate, or sometimes we call it a polysaccharide. We have as we said, they are made of single sugar, right? So we have about 32 plus types of simple sugar, simple sugar. Okay. For the protein, they are made of amino acid. We have about 20 types of amino acid. Okay. For the lipid, we have about 8 types of lipid. By combining all these things together, we can form the cell, different type of cells. Okay. And among them, there are 2 types of language used to assemble all these 68 types of building blocks. These 2 languages are coming from the nucleic acid and amino acid, coming from the DNA, RNA, and the protein. Okay, for the DNA and RNA, for the nucleic acid, we can start looking at the DNA first. There are 4 nucleotides. Okay, and then DNA can use these 4 nucleotides to be transcribed into the RNA. And there are another 4 nucleotides from RNA. Okay? And then the RNA can arrange in the specific sequence, and then those sequence can be translated into the amino acid, and then they give rise to 20 amino acid. Okay, so by arranging the DNA, the 4 nucleic acid in the DNA, we can write the sentences. We can write the sentence for the gene. That sentences can be transcribed into the RNA and then translated to the sequence of the amino acid in the protein. And then the sequence of this protein gives rise to its different property, different shape that will perform functions in the cell. So majorly, there are 2 languages people the nature use in the cell. One is nucleic acid, language. One is the protein amino acid language. Okay, but the protein language actually is coming from nucleic acid language, right? So if we only have for this those few alphabet, this is even less than 26 alphabet, right? From A to Z, it's even less than that. Why? Based on this very few number of the alphabets, we can have very diverse nature, very diverse living systems. The reason is those alphabets can actually organize in very, very diverse way. They have a lot of combinations, okay? And then even after the combination, they can also modify themselves. If you heard, have you heard about the words epigenetics?
Yeah, I've heard of that.
Epigenetics means even the protein is generated by the gene itself can change its properties due to the external environment. Okay, so that change can also lead to different entities, different creatures. So that's the feature of our living system. Okay, so now regardless of epigenetics, even we consider just the combination of those 24 alphabets, we can still appreciate why life is so diverse. So the length of a polymer can vary enormously, from a single one to 1 million one also. Okay, so the number of the possible combinations that it can make the polymer is actually enormous. In the nature, in the biology, for the protein, they are made of the polymer, right? The protein. For the protein, for one single protein, sometimes they can have 1 million, 10 to 6 to 10 to 8 monomers inside. Okay, and then each one can contain many different versions. Okay, let's see. If we have a small polymer, they are not this long. They are not this million monomer long. They are only 15. Okay, we only have 15 monomer in the polymer. Okay, then if we think about the DNA, if the DNA has 4 alphabet, then that piece of DNA, how many versions can it have? Can it have? We only have 15 monomer, and then each monomer have 4 chances, 4 options. Then overall actually is 4 times 4 times 4 and times 15 times, right? 4 to the power 15, right? 4 times power 15 actually equals to 2 to 30. That is already, this number is 1 million possibilities. 1 billion possibilities. Okay, 10 to 9 possibilities. Means if a piece of DNA, it has 15 nucleic acid, that combination can have 1 million possibilities. Okay. Now, on top of that, there's another complexity, another layer of complexity. The structure of the polymer can also change. There's also isomer coming. Okay, so isomer has many different types. First type is called a constitutional isomer. That's also called a structural isomer. For this kind of isomer, you see the top one and the bottom one, these two, they contain the same kind of material, same kind of chemical elements there, but the position of the fluorine is different. Means the structure is different. Okay, even in this case, these two different molecules, they have dramatically different properties. So then they play different functions in the cell. Then they can lead to different cell types. Okay. Also, there are stereo isomer. So that means the spatial, that also called a spatial isomer. Here we list the 3 types of stereo isomer. First one is the mirrored isomer. So in this case, you see these two two types of molecule, they actually contain the same chemical elements, but the structure is different. Spatially, the structure is different. They are symmetric, mirror symmetric. Okay, and because of this symmetric different arrangement, they also have different function. These two also have different function. Okay, so by writing their formula on the paper, they are the same, but in reality, they are different. Structure-wise, it's different. Another one is called cis and trans isomer. Cis and trans isomer is the same thing, same type, same chemical composition, but structurally they are aligned. Spatially, they are aligned differently. For the cis, they actually is the same kind of elements. They are sitting in the same side of the double bond. So double bond is like a plan, and then that plan can be viewed to separate different elements. Now, if the elements, same type of element like hydrogen, they are on the same side of this plan, this double bond, then this is called a cis. If they are on the different side, opposite side of the plan of the double bond, then is in this configuration is called trans. Cis and trans also have different functions. Okay, in the body, they are different function. Okay. For example, in our eyes, when our eyes can can obtain the vision, actually we need some chemical called retina. In our eyes, we use trans retina, not cis. We use trans retina. That retina can bond to our receptor, photoreceptor in our eyes, and then enable us to see the environment around us. Okay, so that's how the function of cis and trans differ. Last one is called ROTAM. This is actually the same chemical components for two molecules, but the arrangement is different for this subunit. In this case, these two are close by. In another, they are close by, they are neighbors. But in the second molecule, they are opposite, in opposite positions. Okay, so then these two molecules also have different functions. So even in the previous slides, we show the same chemical components of the polymer. If they have those isoform complexity, then they also add more variability to the final product. Okay. So yeah, so for the isomer, they have the same number of atoms in the two polymers, but the arrangement of those atoms are different. Now, if we add this layer of complexity in, then for the previous one, we have for previous one n equal to 15 and 4 alphabet. Then if we have only two types of isomer, for example, the bond is straight or bent, two different possibilities, then each one will have two chunks, and then we have n, right? Have n equal to 15, then 2n times 4n, 8n. Then we add together n equal to 15, 2 times 3n, so that's 2 to 45. Then the final number is even more. Okay, if we calculate this, we will get about 10 to 12 numbers of this polymer. And remember, this polymer have 10 to 12 options. It's only a polymer that have the length 15, right? Have 15 monomers. So then if we have a typical macro protein that has millions or billions of amino acids inside, how many possibilities they have, right? Okay, so that's why the fewer alphabet, even we have very little, we don't have many number of alphabet, we can still get very diverse polymers in the nature. Okay, that is tremendous. We can get a tremendous diversity. So that's why in the nature, you see all the plants, all the animals, and all the humans, they are so diverse, right? And then furthermore, throughout the, furthermore, furthermore, throughout the evolution, many of the macromolecule actually is discarded. So eventually, we find on the Earth, there are only these 4 types of macromolecule. And then these 4 types of macromolecule consist of 68 basic building blocks. But even this number is small through the construction, through the reconstruction, modification, and the interaction between them. The cells actually can develop very diverse functions. Okay, and these 4 macromolecules, since they survived through the evolution, they are very robust. So even with these 4 macromolecules, we can obtain most of the functions that we need in the life. Okay, good. Now it's 3:56 now. We can stop here and let's talk in the next lecture. Friday, we have a invited lecture. So that will be excellent. Let's listen to that Dr. Huang's invited lecture. And then we can continue our discussion in the molecular bond and the diffusion. Very good, very good.